Hello aspirants, welcome to Connect IAS. We are back with one more topic, that is DNA barcoding. This we have taken from UPSC PRELIMS 2022 again. So the thing is we are trying to solve the 2022 paper in a more deeper manner. Like we go through the concepts, the important concepts in the question paper and try to solve each and every question in a detailed manner. So that the outcome of this is that we prepare in the from the back, like we are doing the reverse engineering. So we'll look at the concepts, we'll try to understand the concept first, then we'll try to answer the question. So that's how it goes. So on that note, we are looking on one more topic, that is DNA barcoding. So first let's look at the question. The question goes something like this: consider the following statements. DNA barcoding can be a tool to assess the age of plant or animal, distinguish among species that look like, identify undesirable animal or plant materials in processed foods. So, which of the following statements are correct? So, before answering the question, let's look at the concept first. So, what is DNA barcoding? It's basically a technology that allows identification of any biological species by amplifying, sequencing, and querying the information from genic or intergenic standardized target regions belonging to the extranuclear genomes. So basically, it's a technology identifies the any biological species. That's the whole crux of what DNA barcoding is. I mean, here you could see the image. I mean, it is very intuitive. Like these are all the biological species, and DNA barcoding. What it does is it identifies the uh, biological species, the DNA of the biological species, and codes it with a barcode on it. So that's what the DNA barcoding is. So as we have seen, what is the DNA barcoding? Let's see what are the applications of DNA barcoding. It is applied in identification of new species, as we have already seen, and safety assessment of food. Yes, identification and assessment of cryptic species, detection of alien invasive species, identification of endangered and threatened species, linking egg and larval stages to adult species, securing intellectual property rights for bioresources. This is important because still here everything is identification of species. Here, securing intellectual property rights for bioresources as well, and framing global management plans for conservation strategies, identifying plant leaves even when flowers or fruits are not available, identifying pollen collected on the bodies of pollinating animals. So these are all the applications of DNA barcoding. Let's look at once again. Look at them once again. So basically, they are applicable in identification of new species, maybe, and safety assessment of food, identification and assessment of cryptic species, detection of alien species, identification of endangered and threatened species, linking egg and larval stages to adult species, securing intellectual property rights for bioresources, framing global management plans for conservation strategies, identifying plant leaves even the flowers are not available, and identifying pollen collected on the bodies of the pollinating animals. So it talks about identifying the new species, maybe threatened, endangered, everything, anything. And also important is securing intellectual property rights for bioresources and framing strategies, management plan for conservation strategies, and identification of plants and identifying the pollens, etc. So this is all about identifying, that basically done based on the genes. So that is all about DNA barcoding. So now let's look at the question. DNA barcoding can be a tool to assess the age of plant or animal. Is it correct? I think no. Distinguish among species that look alike. Yes. Identify undesirable animal or plant materials in processed food. So the correct options here are distinguish between among species that look alike. Yes. So second one is correct. So option C or D. Identify undesirable animal or plant materials in processed food. So that is 3. So correct option would be 2 and 3. So this is all about DNA barcoding. This is one topic from previous question paper. Let's get back with one more topic, one more interesting topic. So this is all for now. Signing off. For more mind maps, please like, share, and subscribe. Thank you for